Hello everyone, welcome back to channel. In today's video, we are diving into MCQs on prokaryotic gene expression, a crucial topic in microbial genetics. But this video is not just a quiz. I will not only go through each question, but also explain the concept behind the correct answer. That way, you are not just memorizing facts, you are actually understanding the topic deeply. Now, whether you are a student who is preparing for BSc or MSc entrance exam, or you are preparing for competitive examinations such as ASRB NET, CSIR NET, GATE, or whether you are getting ready for NET UG, I hope this video will definitely boost your understanding and your score. In this video, we will cover topics related to gene expression, prokaryotic transcription and translation, positive and negative gene regulation, operon models such as lac operon, tryptophan operon, regulatory proteins. So let us begin with our quiz, and if you find this helpful, don't forget to like, comment and subscribe for more concept based such videos. And also let me know in the comment sections which topics you want next. Let us begin with first question. In prokaryotes, transcription and translation are coupled because A. Both occur in nucleus. Ribosomes can bind to mRNA as it is being transcribed. C. mRNA must be exported before translation. And last option, transcription is faster than translation. So the correct answer is B. Now let us see concept behind it. As we know, prokaryotes, they do not contain nucleus. As a result, their DNA is directly exposed to cytoplasm. Now, as soon as transcription begins, as the RNA polymerase makes mRNA, that process is called as transcription. Our ribosomes can jump onto that formed mRNA and it starts a process called as translation, which is protein synthesis. This transcription and translation processes simultaneously starts in prokaryotes, and this is called as coupled transcription and translation, which is peculiarity of prokaryotic cells. That is why option B is correct. Catalyte repression in E. coli requires which molecule? A. Lac repressor, B. CRP-CAMP complex, C. allolactose, and D. glucose. Again, correct answer is CRP-CAMP complex. Now, why it is correct answer? Now, E. coli prefers glucose over lactose. Lactose is a disaccharide made up of glucose and galactose, whereas glucose is a monosaccharide. As a result, it is simple sugar to get utilized by these prokaryotic cells. And that is why E. coli, which is a bacterium, it prefers glucose over lactose. Now, when glucose is present, lac operon is repressed, even if lactose is available, because it is now having a simple sugar which can be easily utilized. And that is why this control mechanism where glucose is present and that represses lac operon, this process or this control is called as catabolite repression. And key regulator in this catabolite repression is CRP-CAMP complex. Now, what is CRP? CRP stands for CAMP receptor protein, which is also known as CAP. What is this CAP? CAP is catabolite activator protein. And CAMP, it stands for cyclic adenosine phosphate. This is regulatory mechanism, one of the regulatory mechanism in lac operon. Now, what happens here? When glucose is high, it inactivates one enzyme which is called as adenylate cyclase. That enzyme is necessary for formation of CRP-CAMP complex. As a result, when glucose is high, this enzyme becomes inactive and that results into low CAMP. When glucose is low, CAMP rises because now this enzyme is active in form, and this CAMP binds to CRP, which is catabolite activator protein. And why this binding of CAMP to CRP is important? Because this CRP-CAMP complex binds to lac promoter, and it facilitates the binding of RNA polymerase. As a result, lac operon now turns on. When this lac operon turns on, when there is a scarcity or there is no glucose present. When glucose is present, this lac operon will be turned off. The inducer of lac operon is A. Lactose, B. Allolactose, C. Glucose, and D. Galactose. Now students get confused between lactose and allolactose, but the correct answer here is allolactose. Now what is the difference between lactose and allolactose? Allolactose is an isomer of lactose. Now this lac operon, as we seen, has a repressor which is encoded by this lac-I gene, and this repressor binds to operator. Now the inducer here, as stated, is an allolactose. Now when lactose enters, permeases are responsible for entrance of lactose. Now once lactose enters into a cell by permeases enzyme, that lactose gets converted into allolactose, and this reaction is carried out by enzyme called as beta-galactosidase. Now this allolactose binds to repressor, and that repressor falls off. It brings conformational changes into this repressor, and it is now unable to bind to operator. As a result, lac operon turns on, that is, transcription begins. Thus, the inducer in lac operon is not lactose but allolactose. Fourth question, which of the following sequences serves as ribosome binding site in prokaryotes? A. Pribno box, B. Child-Dalgarno sequence, C. Kozak sequence, D. Tata box. And correct answer is Child-Dalgarno sequence. Now let us see concept behind it. Ribosomes in bacteria recognize Child-Dalgarno sequence, which is AGG. It is present upstream of start codon, which is AUG. Now it pairs with 16S rRNA of 30S ribosomal subunit, and it helps to position the ribosome. Now what is this 30S ribosomal subunit? As you know, bacteria contains 70S ribosome. It contains a large subunit which is 50S, S stands for Svedberg unit, and a small subunit which is 30S subunit. In this 30S subunits, 16S rRNA is present, which has important role in phylogenetic analysis. Next question, which of the following statements about operon is false? A. Genes in an operon are transcribed as a polycistronic mRNA. They are common in prokaryotes. Each gene in an operon has its own promoter. And D. Structural genes and regulatory sequences are present together. Correct answer is C. Each gene in operon has its own promoter. This statement is not true. It is false. All the other three are correct in case of operons in prokaryotes. What is operon? It is a functional unit of transcription. One promoter controls all genes, that is multiple genes, and as a result it forms polycistronic mRNA. Example is lac operon, ZYA. These are structural genes. All these structural genes are controlled by one promoter. Next question is, which of the following sigma factors is involved in heat shock response in E. coli? Sigma 70, sigma 32, sigma 54 or sigma 28? Correct answer is sigma 32. Now what is this sigma 32? Sigma factors, these are proteins that guide RNA polymerases to specific promoters to turn on sets of genes, and this sigma 32, which is encoded by gene RPOH, RNA polymerase H, and it has its role in heat shock mechanism. Next question, in tryptophan operon, the attenuation occurs due to high levels of tryptophan, low levels of tryptophan, lack of glucose, or presence of lactose. Correct answer is high levels of tryptophan. Let us see concept behind it. Tryptophan operon has two control levels. One is attenuation and another one is repressor-co-repressor system. In repressor-co-repressor system, tryptophan binds with repressor. As a result, operon is turned off. In case of attenuation regulation, it is based on ribosome movement through leader sequence, which is coded by gene tryptophan L. If tryptophan is high, ribosomes quickly translates leader peptide that forms terminator hairpin. As a result, transcription is stopped. That is, tryptophan operon turned off. If tryptophan is low, ribosome stalls. It halts at that position. As a result, anti-terminator structure is formed and transcription continues. That is, tryptophan operon is turned on. Next question. Which of the following is not a component of bacterial RNA polymerase core enzyme? A. Alpha, B. Beta, C. Sigma, and D. Beta prime. So, as you know, alpha, beta and beta prime, they are the part of bacterial core enzymes, bacterial RNA polymerase core. Whereas this sigma factor, when binds with this core enzyme, it makes a hollow enzyme. As a result, the answer here is sigma, because it is not a part of core bacterial RNA polymerase. Let us see concept. Core enzymes contain alpha 2, beta, beta prime and omega. It is an RNA polymerase, but it cannot initiate transcription. For initiation of transcription, it requires binding of sigma factor. And this core plus sigma factor is now called as hollow enzyme, and that is a functional polymerase enzyme which starts transcription by recognizing a promoter. Next question. Row dependent termination requires hairpin loop followed by uracil rich sequence, row protein with helicase activity, NUS A protein only, or D. ribosome stalling. Correct answer is B. Row protein with helicase activity. As it is row dependent, it will require row protein. In case of row independent, that will require a sequence which forms a terminator in the form of hairpin. So row is hexameric helicase protein. It binds to root site, which stands for row utilization site, which is present on RNA. It uses ATP to chase that RNA polymerase. It unwinds RNA DNA hybrid and it causes termination. Next question. Which antibiotic inhibits prokaryotic transcription by binding to RNA polymerases? A. Chloramphenicol, B. Rifampicin, C. Tetracycline, or D. Puramycin. Correct answer here is rifampicin. What are the mechanism of other three? Here, chloramphenicol, it inhibits the peptidyl transferase, that is translation. Tetracycline, it blocks amino acyl tRNA binding to ribosome, that is again translation. And D, it causes premature chain termination in translation. So all the other three, they have their role in translation, whereas rifampicin, it inhibits prokaryotic transcription by binding with beta subunit of bacterial RNA polymerase. As a result, it blocks RNA chain initiation. It is used clinically in treatment of tuberculosis, that is TB. Next question. Which protein prevents premature transcription termination in E. coli? Correct answer is A. NUS A. Why that is correct answer? Let us see concept behind that. During transcription, RNA polymerase can pause or terminate early. Bacteria have elongation factors that stabilize RNA polymerase and regulate it when it should stop. These are called as NUS protein. N stands for N utilization substances. NUS A has its role in prevention of premature termination. NUS G, it links transcription to translation, that is, it bridges RNA polymerase with ribosome. And NUS B and E, they are important in anti-termination. Next question is, RSC protein in arabinose operon functions as A. repressor, B. activator, both A and B, that is repressor and activator, and D. co-repressor. Here correct answer is both activator and repressor. Arabinose operon, it has ARABAD, which encodes enzymes for arabinose metabolism. Regulator here is ARAC protein. Now ARAC is special because it has a dual role. When there is no arabinose, this ARAC forms a loop and it prevents RNA polymerase. As this way, it acts as a repressor. Now when arabinose is present, this ARAC changes conformation. It binds near promoter and it recruits RNA polymerase. This way it acts as an activator. So it has both roles, as an activator as well as a repressor. Next question. In the lac operon, glucose inhibits A. Lac repressor binding, B. Beta galactosidase activity, C. Adenylate cyclase activity, and D. Permease activity. Correct answer is C. Adenylate cyclase activity. We already have gone through this concept. Next question. Genetic code is said to be degenerate because A. Each codon codes for more than one amino acid. B. There are more amino acids than codons. C. More than one codon can code for a single amino acid. And D. It is universal across all living organisms. The answer here is C. More than one codon can code for a single amino acid. D is also correct, but it itself is another property of genetic code. It doesn't have its role in degeneracy of genetic code. Now what is degeneracy? Degeneracy means more than one codon can code for an amino acid. For example, UUU and UUC, they both code for phenylalanine. Now this is crucial. Why? Because if there are any mutations, the third vowel position or change in the third nitrogen base in codon will not harm an amino acid. That is why this property is important. Now here one codon generally codes for only one amino acid. And there are 64 codons but only 20 amino acids. So option B is incorrect. And A also. Next question. The heat shock response in E. coli is regulated mainly by sigma 32, sigma 70, sigma 28 or sigma 54. Correct answer is sigma 32. Now when temperature increases, protein misfolds and bacteria responds with heat shock proteins. These heat shock proteins in bacteria are expressed by sigma 32, which is encoded by a gene's RNA polymerase H. Next question. The -10 sequence, that is Pribnobox in prokaryotic promoters, is rich in first GC base pair or AT base pairs or GA repeats or GT pairs. Correct answer is B, that is AT base pairs, that is adenine and thymine base pairs. Promoter, it is a region where RNA polymerase binds. It has two key sites, -35 box, which is having a sequence TTGACA, and -10 box, which is called as Pribnobox, and it has a TAAT sequence, which is rich in adenine and thymine. Next question. The consensus sequence at -35 region of E. coli promoter is, we just saw in above question, so correct answer here is B, TTGACA. Next question. Row independent termination of transcription requires A. Hairpin loop followed by uracil rich sequence, B. Row factor binding, C. NUS protein, or D. Poly A, that is polyadenine signal. Correct answer is A. Hairpin loop followed by uracil rich sequence. We saw bacterial termination has two modes, row dependent and row independent. Row dependent require hexameric row protein which has helicase activity, and row independent, it is an intrinsic. It relies on sequence. Next question. Which subunit of bacterial RNA polymerase provides promoter recognition? A. Alpha, beta, beta prime, or sigma? As stated earlier in previous questions, here answer is sigma, that is D. Next question. Rifampicin inhibits prokaryotic transcription by binding to alpha subunit of beta RNA polymerase, B. Preventing ribosome binding, C. Causing premature termination, or D. Inhibiting cellulase activity. Answer is binding to beta subunit of RNA polymerase. Next question, which sequence element is essential for row independent termination? A. GC rich inverted repeat, B. AATAAA, C. Shine-Dalgarno sequence, or D. TTGACA. Correct answer is A. GC rich inverted repeats. Let us look the concept behind it. Now this row independent termination sequence, as said, it is an intrinsic termination. It needs two features. One, uracil rich tail and GC rich inverted repeats. Now this GC rich inverted repeats, as there is a triple hydrogen bond between G and C, it forms a strong RNA hairpin, and this uracil rich tail, it makes a weak RNA DNA hybrid, which results into a halting of RNA polymerase. As a result, process is terminated here. Next question, in the tryptophan operon, attenuation mechanism depends on A. Tryptophan binding to repressor, B. Formation of leader peptide and hairpin structures, C. CRP-CAMP complex, and D. riboswitch. Correct answer is B. Formation of leader peptide and hairpin structures. Let us look concept behind it. Tryptophan operon, as stated in previous question, it has dual regulation. First one is repressor mediated, where tryptophan acts as a co-repressor, and second one is attenuation. It requires formation of leader peptide and hairpin structures. Next question is, which factor is required for coupling transcription and translation in bacteria? A. IF3, B. EFTU, C. NUSG, and D. Rho. Correct answer is C. NUSG. Now what is NUSG? These are the proteins. It codes for protein which binds to RNA polymerase and ribosome simultaneously, and it ensures ribosomes translate mRNA as it is being translated. So this NUSG, it is a gene which codes for protein that facilitates the coupled transcription and translation in bacteria. Next question is, Shine-Dalgarno sequence in prokaryotes is complementary to A. 5S rRNA, B. 16S rRNA, C. 23S rRNA, or D. tRNA anticodon. Shine-Dalgarno sequence, as stated in previous question, it is ribosome binding site on mRNA, and it has its complementary sequence in 16S rRNA, which is present in 30S ribosomal subunit. Next question, which of the following is not required for prokaryotic transcription initiation? A. Sigma factor, B. Core RNA polymerase, C. Promoter DNA, and D. Rho factor. Correct answer is D. Rho factors. So transcription initiation, it requires core enzyme plus sigma factor, and it also requires DNA promoter, but it does not require Rho factor. Rho factor has its role in termination, not in initiation or elongation. Next question, which antibiotic inhibits translation by binding to 30S ribosomal subunit? Chloramphenicol, streptomycin, rifampicin, or erythromycin? Correct answer is B. Streptomycin. Streptomycins, these are antibiotics produced by Streptomyces species. Antibiotics, they targets ribosome differently. Now streptomycin, it binds to 30S ribosomal subunit, as it causes misreading, while chloramphenicol and this erythromycin, they binds to 50S subunit. Even though both have their target of 50S subunit, they work differently. Chloramphenicol blocks an enzyme called as peptidyl transferase, whereas erythromycin binds to 50S subunits and blocks translocation, that is movement of ribosomes, whereas rifampicin, as stated previously, it inhibits RNA polymerases and not ribosome. So you need to carefully study this mechanism of antibiotics and where they attack on prokaryote. Next question, which sigma factor is used in nitrogen starvation response? A. Sigma 70, B. Sigma 32, C. Sigma 28 and D. Sigma 54. Correct answer is D. Sigma 54. Now, sigma 70, it is a housekeeping factor. Sigma 32, it has its role in heat shock mechanism. Sigma 28, it has a flagellar genes and it helps in movement of bacteria in chemotaxis. Whereas this sigma 54, it has its role in nitrogen metabolism. Next question, which of the following is not a component of bacterial RNA polymerase core enzyme? Alpha, beta, sigma or beta prime? We have already seen this. Correct answer is C. It is sigma. Next question, enzyme responsible for charging tRNA with amino acids is A. Peptidyl transferase, B. Amino acid tRNA synthetase, C. EFTU or D. Ribosomes. Correct answer is B. Amino acid tRNA synthetase. Translation, it requires amino acylated tRNAs. It is done by amino acid tRNA synthetase enzyme. There are around 20 types of this enzyme and 1 per amino acid is required. It is highly accurate and it has proofreading activity. Next question, which of the following is not true about lacoperon? Now let us see these options one by one. It is inducible, that is correct. Here you have asked which of the following is false, that is not true. Allolactose acts as inducer, this one is also correct. It is controlled by both negative and positive regulation, this is also correct. Glucose presence enhances its expression, this one is false. Because we have seen glucose when present, it is preferred over lactose and it reduces the concentration of CAMP. As a result, lactosoperon is turned off. So here answer C is not true in case of lacoperon. Next question, which enzyme unwinds the DNA during transcription in prokaryotes? A. DNA helicase, B. Topoisomerase, C. RNA polymerase itself or D. DNA gyrase. Correct answer is RNA polymerase itself. DNA helicase has its role in unwinding but not during transcription. You are asked for during transcription. It has its role in unwinding DNA but during translation. That is why this one is incorrect option. Topoisomerases and DNA gyrases. Now these two enzymes, they relieve torsional stress in both replication as well as translation. But they are not directly involved in unwinding of DNA for polymerase activity. That's why these B and D are also incorrect options. Whereas this RNA polymerase, it has both the helicase activity as well as polymerase activity. And that is why this C is correct answer. Next question, which factor facilitates row dependent termination in bacteria? A. NUS-A, B. NUS-G, C. Sigma 70 or D. IF2. That is initiation factor 2. Correct answer is B. NUS-G. Row dependent termination requires row factors and these are encoded by NUS gene. It enhances rows access to RNA polymerases. Without NUS-G, termination efficiency decreases. So it helps in facilitating this row dependent termination. Next question, which sequence acts as a ribosome binding site in bacteria? A. Kozak sequence, B. Pribnobox sequence, C. Shine-Dalgarno sequence or D. TTGACA. Correct answer is C. Shine-Dalgarno sequence. Kozak sequence is a ribosome binding site but in eukaryotes, not in prokaryotes. That's why A is incorrect option here. Next question, which subunit of prokaryotic ribosome contains peptidyl transferase activity? A. 30S subunit, B. 50S subunit, C. 16S rRNA or D. Sigma factor. Answer is B. 50S subunit. It contains peptidyl transferase activity. And more specifically, if there is option of 23S rRNA, so that will be a more correct answer. Next question, which initiation factor brings formylmethionine tRNA to ribosome in prokaryotes? A. IF1, that is initiation factor 1, B. Initiation factor 2, C. Initiation factor 3 and D. EFTU. Correct answer is B. Initiation factor 2. Initiation of translation in prokaryote requires 3 different initiation factors. So IF 1, 2, 3. They are involved in translation initiation, where initiation factor 1 prevents premature tRNA binding. Initiation factor 2, it brings formal methionine tRNA to P site, and initiation factor 3, it prevents premature association of subunits. Next question. The first amino acid in bacterial protein synthesis is A. methionine, B. N-formyl methionine, C. valine, or D. isoleucine. Correct answer is formyl methionine, that is B. Whereas methionine, it is a first amino acid in eukaryotes. Prokaryotes uses formyl methionine, that is N-formyl methionine, as initiator amino acid. Eukaryotes uses methionine. Next question. EFTA is bacterial translation which delivers formyl methyl tRNA or charged tRNAs to A site, C. ribosome subunits, or D. mRNA to ribosomes. Correct answer is B. Charged tRNAs are delivered to A site by this EFTU. These are elongation factors. EFTU, it is GTP dependent and it delivers amino acid tRNA to A site. EFTS, they recycles EFTU. EFG, in these elongation factors have their role in promotion of translocation. Next question. During translation elongation, peptidyl transferase activity is catalyzed by A. protein enzyme, B. rRNA, C. tRNA, and D. EFG. Correct answer is B. rRNA. Why this rRNA is correct answer? Ribosome, it is a ribozyme and not a protein enzyme. In this 23S rRNA, that is in larger subunit, it catalyzes peptide bond formation. This activity is intrinsic to rRNA and not tRNA. That is why this rRNA is the correct answer. Next question. Translocation step in prokaryotic translation requires, we already have seen this in previous question, A. EFG and GTP, B. EFTU and ATP, C. IF2 and GTP, and D. RF1 and GTP. Correct answer was EFG and GTP. Next question. Which release factor recognizes UAA and UAG stop codons in prokaryotes? Release factor 1, release factor 2, release factor 3, or EFTU? EFTU is incorrect answer because it has its role in elongation. Now out of these 3 release factors, let us see one by one. Release factor 1, it recognizes UAA and UAG. Release factor 2 recognizes UAA and UGA. And release factor 3 helps in dissociation of release factor 1 and release factor 2. That is why correct answer is A. Release factor 1. Next question. Which of the following is a riboswitch? A. mRNA region binding small metabolites. B. Protein that binds mRNA. C. Regulatory sRNA. And D. DNA operator sequence. Correct answer is A. mRNA region binding small metabolites. Now what are riboswitches? Riboswitches are regulatory segments in mRNA. They directly bind metabolites. Binding changes mRNA secondary structures, which ultimately affects transcription and translation. Next question. Polycistronic mRNA is characteristic of A. Prokaryotes, B. Eukaryotes, C. Archaea only, and D. Viruses only. Correct answer is prokaryotes. Next question. Which sigma factor controls expression of flagellal genes in E. coli? A. sigma 28, sigma 32, sigma 54, or sigma 70. As already seen, it is sigma 28. Next question. Which of the following operon is repressible? A. lacoperon, B. arabinosoperon, C. tryptophan operon, and D. maltosoperon. Correct answer is C. Tryptophan operon. What are repressible operon? They are normally on and turned off when product is abundant. What is product in tryptophan operon? Tryptophan. It is anabolic mechanism. Whereas this lac operon, arabinose operon and maltose operon, they are indicible systems where presence of these products turns that operon on. In tryptophan operon, tryptophan, it acts as a co-repressor as it binds to repressor and it turns off the operon. So when product is abundantly present, the repressible operon is turned off. Whereas when product is abundantly present and operon is turned on, that operon is called as indicible operon. Next question, which of the following is an example of attenuation? A. Lac operon regulation, B. Tryptophan operon regulation, C. Arabinose operon regulation and D. Maltose operon regulation. Correct answer is B. Tryptophan operon regulation. We already have seen this concept. Next question, which of the following is not an elongation factor in prokaryotic translation? A. Elongation factor Tu, elongation factor Ts, elongation factor G and I of 3. Correct answer is D. I of 3. Why? Because it is an initiation factor and not an elongation factor. Next question, in the lac operon CRP-CAMP complex binds near A. Operator, B. Promoter, C. Terminator and D. Leader sequence. Correct answer is it binds upstream to promoter. CRP, it requires a CAMP. This complex binds to upstream of promoter and it recruits RNA polymerase. Why that complex is important? Because it ensures positive regulation when glucose is absent. Next question, which release factor recognizes UGA stop codon in prokaryotes? RF1, RF2, RF3 and EFG. EFG is incorrect. Why? Because it is an elongation factor and not a release factor. Now out of these three, if you recall, we already have seen all the three release factors and the correct answer here is release factor 2. It recognizes UGA stop codon. Next question, which of the following is true about prokaryotic transcription and translation? So correct here is C. They occur simultaneously. Why? Because transcription and translation are coupled in prokaryotes. Why they are coupled? Because there is no nucleus. As a result, when mRNA is produced, ribosome binds on it and it starts translating that mRNA. This is called as coupled transcription and translation, which occurs simultaneously in case of prokaryotes. Now last question, in bacterial gene regulation, catabolite repression ensures A. Glucose is used preferentially, B. Lactose is always metabolized first, C. All operons are constitutively expressed and D. Translation stops early. Correct answer is A. Glucose is used preferentially. This is a role of catabolite repression. What is catabolite repression? It is a global control mechanism. It ensures glucose is used first even if the alternative sugars are present, and it is mediated by CRP complex. So this was all about a quiz on prokaryotic gene expression. I covered concepts as much as possible. If you find this helpful, let me know in comment section. Thank you.